to introduce Nicole Dubilier. We've known for a very, very long time. We actually spent quite a bit of time together in the same lab. And I hope I can say that we've been very good friends over the years. And it's a real pleasure to have her give a keynote on a relatively simple microbiome today on Symbioses. But she's taken this system really very, very far. And I would also say with sometimes some daring approaches that combine a lot of creativity, environmental work, as well as sophisticated genomics and proteomics. And finally, I have to say that she's a director at the Max Planck Institute for Marine Microbiology in Bremen, where she leads the Symbiosis group. And with this, Nicole.
Many thanks, Martin, for your introduction and for your saying that we're still friends after having shared a lab together many, many years ago. Martin and I both worked on the chemosynthetic symbioses that I'll be talking about here, and I'll be focusing on the mussels that you see here at hydrothermal vents. They were discovered only 43 years ago. So Martin and I both worked on these symbioses in Colleen Cavanaugh's lab at Harvard, and she was doing her PhD when they were discovered. So it's still a generation there that worked on them and is still active. And they completely revolutionized our understanding of the energy sources that fuel primary productivity on Earth. I say this was the last major discovery of an ecosystem we didn't know anything about, and that's kind of it for at least planet Earth. And the productivity is tremendous. It's as large, the biomass, as that of tropical rainforests, coral reefs, and kelp forests. And what is so amazing is that the majority of this biomass is based on microbe animal symbioses. And the way that they function is that the symbionts use reduced inorganic compounds such as sulfide to feed their hosts via CO2 fixation. So they're the primary symbionts for their hosts. When you think about photosynthesis, the other form of primary production, what you have is the light energy that is fueling the chloroplasts, which are also symbionts, or derived from cyanobacteria symbionts. And they're fixing CO2 into biomass, and that's feeding the plant. And in chemosynthetic symbioses, you've got chemical energy. It's the oxidation of reduced compounds such as hydrogen sulfide or methane that is being used by the symbionts inside of the animals to fix CO2, the energy from the oxidation of the reduced compounds, to fix CO2 into biomass and to feed their animal hosts. They were first discovered, these symbioses, at hydrothermal vents in the deep sea off the Galapagos Islands in 3,000 meters depth. And then one went on to discover that they occurred at cold sweeps in the Gulf of Mexico. We now also know that they occur in whale falls. But what is really remarkable is that there is a tremendous diversity of these symbioses in shallow waters sediments, in seagrass beds, mangrove swamps, coral reef sediments. And zoologists for many, many years had wondered why they found animals with a reduced gut or that had reduced their digestive system entirely. And it was assumed they simply lived off of organic material in the sediments. But we now know that they too have chemosynthetic symbionts. And they have evolved these types of symbioses numerous times in evolution, independently of each other. At least 15 bacterial groups have been able to establish these symbioses. At least 2 protists and 9 animal groups have established these symbioses. And what that indicates is that there's a very strong selective advantage for both partners, for both the bacteria on the one side and the eukaryotic hosts on the other side, in coming together to form these symbioses. These are some of the hosts that we work on in my group. I've actually cheated a little bit because the placozoans are not chemosynthetic. They have different types of symbionts, but we've been doing some very cool research on them, and so I wanted to include them. And today in my talk, I'm going to focus on the work that we've been doing in the deep sea, on deep sea mussels. And so you may ask yourself, why would anybody study symbioses in organisms that are not genetically tractable and can be difficult to collect? And indeed, this is something, at least when I was writing my first proposals, that I would continuously get as a criticism. And I think all of us that are attending this meeting, but it's amazing when you go away from a crowd that is, let's say, not as broadly focused as we are, many different systems, that we really need to go out into nature and look at the broad diversity of what is out there. And the reason that we're able to do that now is the fantastic tools that we have. They were discovered hydrothermal vents because because submersibles were available with which scientists could go to the bottom of the sea, but we now have a wealth of other tools that are helping us study these non-cultivable organisms. And as Martin mentioned, we use a broad array of tools in my lab, everything from in situ experiments to high resolution imaging and incubations in the lab, in the field, and a whole bunch of omic toolboxes. As I said, I'm going to focus in on the deep sea mussels that we work on, and I am going to emphasize that the symbionts, as yet that they have these mussels, remain uncultivable, but we can keep the hosts in aquaria. And so that has been really helpful for some of the experiments that I'll be describing. I'm going to present some of the imaging work we've been doing, and it's a fantastic team that has been doing this work. Nicole Dubilier, a postdoc in my lab who did his PhD in the University of Vienna, has been working with me, and two students that have just gotten their PhD, Benedict Gaia, who does a lot of MALDI, metabolite imaging, and Max Franco, who's still working on his PhD. So the symbioses in deep sea mussels are low diversity, high abundance microbiomes. What you see here is a micro CT image of a mussel, and the symbiotic organs are its gills, colored in yellow here. And we estimate that in a mussel of about 7 centimeters size, that there are 10 to the 12th bacteria cells in a single individual. And the community is made up, as a rule, of only 2 bacterial species, a sulfur-oxidizer and a methane oxidizer. So that means that the omic tools that we use make it, give us an advantage over the high diversity systems that many others are working on. The symbionts live intracellularly inside the gill tissues, and the gill tissues consists of these rows and rows and rows of filaments. And these filaments are very similar to, you imagine a vertical slatted curtain, and imagine that each slat is one of these filaments, and that there are thousands and thousands and thousands of these slats behind each other. And each of these slats, what you see here, a slat or a filament, are 2 filaments in cross section, and each of them houses the bacterial symbionts. This is using 16S probes that are specific to the 2 symbionts, the sulfur-oxidizer and the methane oxidizer. And you can see that they are inside the gill cells. I'll show you an electron microscopy image that they're actually inside. Another thing that you can see here using these probes is that the outer edges of the gills, the ciliated edges where they're streaming the water along their gills, they do not contain symbionts. And so despite the fact that this is the same tissue and these cells are right next to each other, some of them contain symbionts and some don't, showing a very clear specificity at the cellular level of these associations. And this is a transmission electron micrograph of a single host bacteriocyte, a single gill cell that houses the 2 different types of symbionts. You can see the larger ones are methane oxidizers, the smaller ones are the sulfur oxidizers that are housed inside these bacterial gill cells. And when you think about, when I thought about how I can make this hopefully interesting to those that are working on epithelial tissues, Gill cells are epithelial tissues. They're like intestinal epithelial. If you compare it to the human gut, what you also have is a single columnar epithelial cell lining, which is exactly what we have in the Gill cells. And the gills are an inside-out gut in that the apical ends are towards the seawater. Here in the human gut, they're towards the intestinal lumen. Here they're reaching out, and this is where the seawater is being streamed past their gills. So we all know that hosts adapt their body plans in response to their microbiome. This can be short-term responses, but in many of the chemosymbiotic associations, we see adaptations that have occurred over millions of years. And what we see in the mussels here is a massive enlargement of their symbiont-containing gills. So what you see on the left here is a micro-CT of a non-symbiotic shallow water blue mussel Bathymodiolus. This is one of its gill epithelial cells, very flat, lots of microvilli, I'll come back to that, and then very narrow or normal sized gills. And when you look in a cross section now through one of these, I'll call them slats or filaments, you can see how gigantic they are. They're hypertrophied. And that's because each single cell that is sitting inside of one of these filaments is filled with a symbiotic bacteria. The way the symbiosis functions is that the Gill cells are modified to eat the bacteria. So they eat their symbionts through phago-lysosomal digestion. And all these black spots that you see here in the basal edges of the epithelial cells are not pore staining. They're actually gigantic phagolysosomes, and conveyor belt like the symbionts are in the apical part of the cell. They're dividing there, they're growing, and they're being digested in the basal part of the host cell. Other body plan adaptations that we see in these mussels is a simplification of the gut. This is again the shallow water Mutilus that we always use for comparisons. And what you see here is the digestive system, and focus here on in red, the intestines of the shallow water mussels. And you can see that they're looped and turned around. And the reason for that is because they're digesting phytoplankton, and that's not easy to digest. And so they need a rather complex gut system so that they can digest the food. In the deep sea mussels symbiotic, in the larval stages that also feed on phytoplankton, they actually go all the way up to the surface. We have some very, very cool new evidence that they go from the deep sea all the way up to the surface to feed and then go back down to the deep sea. And as they settle on the sea floor, their intestine straightens out because they are no longer are feeding on this hard to digest food, but are rather feeding on their symbionts in their gill cells. The symbionts can be lost and regained, and we know that because we've been keeping them in aquaria in Kiel together with our colleagues from GeoMar. We collected mussels in the Gulf of Mexico in 2014. We kept them in aquaria starting in 2014 for 1 year and now for several years. And when we started, we kept them at 10 micromolars methane, which we thought would be more than sufficient in a constant flux at least to keep them happy. And I'll show you what happened to them after 1 year. So this is what a single host bacteriocyte, a gill cell, looks like when you sample the mussels freshly from the field. It's full of the symbiotic bacteria that you can see inside the host tissues. This is what all our poor starving mussels looked like after 1 year of keeping them in the aquaria. They didn't have a single symbiont. We couldn't find a single one. So we ramped up the methane concentrations to 40 micromolar, and what you can see is that they were able to regain their symbionts, very similar to corals and the loss and gain of symbionts. We can see this also in our systems. What we also see is something that is quite well known from pathogenic bacteria, which is that the microvilli on the surface of the cells that host the symbionts are lost. Again, here, a host cell, a Gill cell, where there are no symbionts, and you can see the microvilli, which are typical for all epithelial cells. And we know now from looking at larvae, as soon as a single bacteriocyte enters one of these cells, they lose the microvilli. This is called microvilli effacement, and it's been well studied in pathogenic E. coli bacteria, where where the bacteria invade the cells and induce loss of the microvilli. As a final, this is brand new. This is something that we've actually just started to analyze the data from, which is Nicole Dubilier noticed that when we looked using electron microscopy, when we looked carefully at single bacteriocyte cells, we noticed very often that there were openings at the apical end of the bacteriocyte to the outside, this is where the seawater is streaming by, and the arrows show these openings. But we were never sure if these were artifacts of poor fixation. We weren't let's say, after seeing them for many, many years and continuously, we started to think they could be real. The problem, though, in imaging these kind of vacuoles is that you want to actually be able to section through an entire cell in not only in the XY axis, but also in the Z axis through the depth. But your resolution when you're doing electron microscopy and you do a section is as great as 70 nanometers, so very great. And there's a wonderful method out there called focus ion beam scanning electron microscopy, FIB-SEM, that has been developed over the last 10 to 20 years and is increasingly being used for biological samples. And we were very lucky to get time to use these instruments for a couple of days together in a at the EMBL, the European Molecular Biological Lab in Heidelberg, with Yannick Schwab and Nicole Schieber helping us with the samples. And what I'll show you next is a sample through a single bacteriocyte. What you're looking at is a single Gill cell in which the symbionts sit. This is a reconstruction going nanometer for nanometer through the top of the cell through to the bottom. We're looking at the top end of the cell, and here all those red dots that are colored are the actual openings that we saw in the 2D analyses, and we don't see them in symbiont-free host cells next to them. So it's very clear an adaptation of the cells to their symbionts. And now what you're going to see is what it looks like in a 2D section where you can see these openings that are allowing the symbionts to sit inside the cell, but they're open to the outside environment where all the nutrition that they need, the reductants, the sulfide, and the oxidants are entering inside the cell. Want to continue. From here on in, I'm going to now switch and tell you a little bit about our omic analyses. And these were done, a lot of the results that I'll be showing you, by another fantastic team, Jill Peterson, who now has her own lab at the University of Vienna, and Rebecca Anzaga and Liz Zayavedra, who have now gone on to do postdocs at the Quadram Institute in Norwich. We've talked a lot about strains, but what we haven't really discussed is how difficult it really is to analyze strain diversity properly. In cultivable microorganisms, Martin has done wonderful work, particularly because he's put a strong emphasis on not letting the bacteria that he cultivates, the Vibrios, sit in the lab too long, because use it or lose it. Anything you cultivate for many generations on a certain medium is only going to, as a rule, keep the genes that it needs to survive in the medium you're culturing it in. And so, as we've heard also in the session before, many of us are now using metagenomic methods. We tried for quite a while to use single cell amplified genome methods, but overall, I have to say that the tools that are now available for metagenomic sequencing are so excellent with short read and long read sequencing that I feel that that is really the way to move forward in understanding strain diversity. One of the things we've been arguing about in my lab is what actually is a strain in terms of metagenomic analysis. And I was extremely happy to see this wonderful review. I'd like to point out that Thea van Rossum, together with Per Bok at EMBL, recently published in Nature Review Microbiology. And it's very important when most people just think anything underneath a species is a strain, but it really isn't. And defining what is a taxonomic strain or a natural strain is something that we've been thinking about a lot in my lab. And I'll remind you, and this is important, that when you are doing 16S sequencing, no matter whether it's PacBio long read sequencing, you are staying at the species level at best, and that you really need to do higher resolution sequencing, whether it's metagenomic or genomic sequencing, to get down to the level of resolution that is interesting. And I'll show you why we think that's really important and why it matters. And many of you have already shown strain diversity and the importance of it. We've been looking very carefully at the symbionts, only the sulfur-oxidizing symbionts. I'm only going to focus on them now, that occur in mussels in the Atlantic on the so-called mid-Atlantic ridge, which is where the ocean plates are spreading apart, and there are many hydrothermal vents along this mountain range at the bottom of the ocean. And there are 4 Bathymodiolus species that occur there. They're very closely related, and based on 16S, each of them has a specific symbiont, a specific phylotype. But as we know, the resolution of 16S is not high, and I will also remind you that the divergence rate of 16S are extremely conservative. We estimate that it's somewhere between 5 and 50 million years before you see 1% sequence divergence, which is often the resolution of many other studies. And so one of the things that Rebecca did during her PhD was look at mussels that we had collected along the mid-Atlantic ridge. And one of the first things she did to look at their, looking at their metagenomes of the sulfur-oxidizing symbionts was the number of SNPs that she saw in each single mussel individual. And what you see here are the SNP analyses. And what she found was a SNP frequency that was quite similar at each site, but varied between sites between 5 and 11 SNPs per 1,000 base pairs. And so one of the first questions I had, Rebecca started this in 2016, this work, and it was still, there weren't that many methods out there for reconstructing strain diversity based on metagenomic Illumina sequencing. Is that a lot or a little? And so when you compare the diversity that you see, the SNP number that you see in true intracellular symbionts like the Bathymodiolus, and you see that SNP diversity is very low. And then the SNP diversity that we are seeing in the mussels is very similar to that in the gut microbiome. And Rebecca spent quite a while trying to reconstruct the actual strain diversity inside the mussels inside each individual, using both Illumina and PacBio sequencing and haplotype reconstruction based on a method that was actually developed for viral strain reconstruction called ViQA-S, essentially using many of the methods that others are using with SNP linkage and frequency. And she tested this method also with 16 or 17 E. coli strains to see if she could reconstruct also the number of strains in them. We're quite confident that the number of strains are correct. And what she found is that as many as 16 strains per host individual of sulfur-oxidizing symbionts co-occur in a single individual. And when we first presented this work at an evolution meeting, they told us, well, that can't be what you're seeing, because evolutionary theory predicts that in intimate intracellular symbioses, these strains are competing with each other for space and resources to the detriment of their hosts. And in fact, Tammy Lieberman and Arilyn Conwell today were showing beautifully that there is no strain diversity in the pores. It reminded me a lot of what we're seeing here in the host cells. So we've come up with some hypotheses that can explain this diversity, but let me first explain why we think this diversity matters and what it results in. The strains vary in their functional repertoire. And what Rebecca did is created for each site where we sampled mussels, a consensus sequence from all the mussel individuals, and then mapped the metagenomes of the symbionts from single individuals onto the consensus sequence. And so if a gene is present in all the reads that you find from the community of sulfur-oxidizing bacteria in a mussel, then you would expect the coverage to be fairly high, up to 100%, as you see here in the graph. But there are other strains will only be present, other genes that will only be present in some strains, but not in all. And what Rebecca then did was looked at the so-called lower coverage to see which genes were specific to strains. And these are the genes that she looked at more carefully. And as always, about 80% couldn't be annotated or clearly defined, but we lucked out with the 20 to 25% that we could annotate. Many of these were genes that we feel provide a strong, or that we are convinced provide a strong selective advantage for the symbiotic bacteria: virus defense genes, toxin-related genes, and metabolic genes. And I will emphasize that these genes are expressed. We see them in the transcriptome and in the proteome. And it's also intriguing to see that many of these low coverage genes have been acquired through horizontal gene transfer, which we see both based on the nucleotide usage patterns and phylogenetic reconstruction. And here's a summary of some of the genes that you see, and many of them are involved in recognizing bacteria at the surface of a cell, phage defense genes, but also key genes in the metabolic pathways of the symbionts. For example, the genes that the symbiotic bacteria can have to use hydrogen as an energy source. And I'll briefly explain what we saw only here in the variability of using hydrogen as an energy source. So until about 10 years ago, it was assumed that there were only two energy sources that can really fuel chemosynthetic symbioses: hydrogen sulfide and methane. And we were able to show in 2011 that hydrogen is an energy source that can be used by the symbionts, and it's the sulfur-oxidizing bacteria that have acquired the genes through horizontal gene transfer to use hydrogen as an energy source. And what Rebecca did was looked at the distribution of the symbionts that have these genes that enable them to use hydrogen. And when she first showed me this graph, I got goosebumps, because when I was first doing the experiments to see if the mussels can use hydrogen, I was doing physiological experiments with the mussels, where I was incubating them in little vials with hydrogen and then looking after 24 hours if they could take it up or not. And I did these experiments at this vent site called Semenov, and that has incredibly high hydrogen concentrations. And you can see that 100% of the strains that are present in the mussels there have the genes that allow them to use that. And similarly for another vent with high hydrogen concentrations. But then down here, Clueless, hydrogen concentrations are very low, and only about 20% of the strains within single mussel individuals can have the genes that allow them to use it. And the goosebumps came from the fact that we could have had the cruise to Clueless, and I never would have been able to have really shown with the first experiments that they use hydrogen as an energy source. Okay, and Lucky Strike with intermediate hydrogen concentrations. We care a lot, as I have already shown, about visualizing our symbioses, and Miguel Gonzalez Puros has been doing excellent work in combining a general probe, a 16S probe for all the sulfur-oxidizing symbionts present in our symbioses, with a probe that is specific for the gene that allows the symbionts to take up hydrogen as an energy source. And what you can see is that there's a clear stratification in the Gill filament, with some of the host cells full of strains that can use hydrogen as an energy source, while others are not. So this is what we are now arguing in summary of the advantages of strain diversity. The mussels are continuously exchanging their symbionts, both in the environment, and they're exchanging them with co-occurring hosts at the hydrothermal vents and with free-living bacteria in the vent environment. And they're doing this during the lifetime of a host. They can take up the symbionts continuously and lose them. And this allows them to associate with the strains that are best adapted to the vent environment. And that adaptation is rapid, and it's rapid to the local conditions in which they are. So within the lifetime of single host individuals. And so, in contradiction to the theory that predicts that co-occurring strains will be competing with each other, what we are arguing is that doesn't hold true for associations in which symbionts acquire their resources from the environment. And even though gut microbiota are not intracellular, they're getting their nutrients, their resources from the environment when you eat, which is why we also see this strain diversity. And in nutritional symbioses, competition between strains can be extremely beneficial for hosts, because the dominance, they will be competing for dominance of those strains that can best transform external resources into biomass. And with that, I'll finish my presentation. I didn't time myself. As Martin said, I do try to speak or I do speak quickly. This is a fantastic group, my lab that we've been working with, and the funding sources that have allowed us to do this work. And I thank you for your attention.
Hey, great talk, Nicole. Thank you so much. We do have a couple of questions, but I want to start off with one that I really wanted to ask, because it's a little bit puzzling to me that you see this variation. If there's some hydrogen present, at least, is there a trade-off to having a hydrogenase?
Well, okay, I'll generalize. First of all, I think what we are seeing is that use it or lose it. Indeed, that carrying extra genes around is definitely not advantageous if you're not going to use those genes, if it's not beneficial. That's what we assume. All hydrothermal vents have hydrogen. It's actually the concentrations of hydrogen that matter.
Right, right. But I would imagine that even if you have a little bit of hydrogen and there's no disadvantage to carrying the hydrogen,
I see what you're saying,
that you wouldn't actually lose it, because you will always gain a little bit of a growth advantage at least.
Yeah, so then we've discussed the energy that you get from using sulfide as an energy source versus the energy you get from using hydrogen as an energy source. And although the redox potential is high for hydrogen, overall, our estimates are that you can fix more CO2 with sulfide than you can with hydrogen. So there has to be a trade-off where hydrogen concentrations become so high where it becomes beneficial. Does that make sense? In other words, depending on, it's just like the human gut, depending on whether you eat a lot of chocolate or not, you're going to get a lot of chocolate eating microbes.
Okay, thanks. Yeah, we have a bunch of questions from the question and answer tab here, and I'll try and read it. It's incredibly small on my screen. The first one is from Handu Shi, and she's a postdoc from Stanford. And she says, it's really cool to see that modulating methane concentration could modulate symbiont levels. At the low concentration of methane, it looks like all the symbionts in the gill were lost. in that case, what is the reservoir for the symbiotic bacteria that allowed reseeding at higher methane concentrations?
Yeah, that's a very nice question. Thank you. The aquarium in which they are sitting in is the seed bank. And one of the first questions we had was whether they are taking up the exact same symbionts they had, or whether the methane in the seawater also allows free-living methane oxidizers that are there to grow. And this is seawater that is coming from the Baltic Sea. And so there's a high diversity, really, of methane oxidizing bacteria in there. And it's fascinating to see that they are taking up their exact same symbiont. Exact same, I have to be really careful because we still want to do strain analyses down to a level where I really believe it. But certainly at the 16S level, we cannot distinguish them.
So the next question is from Anthony Shiver, who is also a postdoc. And he's asking, does spatial structure of the two symbionts play an important role in the system? From the fish images shown at the beginning, it seems like the two species were spatially separated. Do they both need to be in a cell at the same time in order to perform their chemistry?
So the two types of bacteria, sulfur-oxidizers and methane oxidizers, co-occur in single host cells. And in fact, we're seeing some very, very cool metabolic interactions between these two types of bacteria. The first product that a methane oxidizer produces is methanol. And many methane oxidizers actually leak out methanol. It's well known from plants, which also have methane oxidizers and produce quite a bit of methanol. And another gene that is horizontally transmitted that we see in the sulfur oxidizers is one that allows them to use this methanol. So there's metabolic co-occurrence going on. In terms of strain diversity, that's one of the most for us, one of the things we're very excited about doing. We spent 3 years trying to actually get the single host cells to disintegrate to the point where we could do single host cell sequencing, because prediction, it is possible that each single host cell hosts only a single symbiont strain, very similar also to what Errol and Conwell were showing, which is a combination of the first cell that colonizes it, how it divides, and how successful it is to outcompete any other co-colonizing strain. And these are questions that we'd like to look at the single host cell level, but just getting these frigging gill dishes to disintegrate has been a nightmare.
Okay, thank you. So Carlos Vogt, who's at the EMBL in Heidelberg, says, thank you very much for the wonderful talk. Regarding the strains in mussels from hydrogen-rich sites, have you considered to bring mussels from hydrogen-rich sites to hydrogen-low sites to study the contribution of symbiont hydrogen usage on the host? Thanks.
Big no-no. So we used to, way back when hydrothermal vents were discovered and there was no code of conduct about what you were allowed to do in the deep sea, researchers very happily took for example, the tube worms from one site and moved them to another site and transplanted them to actually answer exactly the question you're asking. But the code of conduct has agreed that this is not good to do this, and so we no longer do that. What we have done, though, and this is a set of experiments I'm very excited, we're analyzing them at the moment, is we've taken the mussels and we've moved them away from the vent sites. So we moved them only 100 meters away. We put them in cages. There's no longer the substrates they need. So it's very similar to the starving in the aquaria, but we do it in the deep sea. And then we've developed a method that allows us to take RNA later down to the bottom of the seafloor and actually fix the mussels at the bottom of the seafloor in RNA later, and then look at their expression and how they change in adaptation. So we call that we can do removal experiments, but real transplant experiments is not allowed according to the code of conduct.
Okay, so Alex Christoph is a grad student from Berkeley and has a short question. Do you observe any recombination events between strains within mussels?
So unfortunately, we can't resample the same mussel. Every time we sequence a mussel, that's it, it's dead. And so that's a very hard question to answer. What I can say is, and you might have seen that in some of the electron micrographs, is that we see a tremendous formation of outer membrane vesicles. The little, if you saw around the bacterial symbionts, there's a ton of them. And in fact, when we wanted to look at the phages inside the symbionts, what we collected was a ton of OMVs. And we see the genes being expressed that you need to make these outer membrane vesicles. And so it's well known that that's one of the mechanisms for horizontal gene transfer. And yes, we predict that that's how it's being exchanged. That is one possible mechanism of how genes could be exchanged.
Okay, so then we have Elizabeth Hughes, who's a postdoc in the Valdivia lab at Duke, is saying, so interesting to learn about this symbiosis. Did you use the HyDB database for classification of the hydrogenases? Also, what determines the hydrogen concentration at these vents?
So I don't know, I would have to ask Rebecca, who's not here, which database she used. What I do know is that we looked at the closest, that we simply blasted the genes against the databases that were there and created our own database. So I'm not sure if the HyDB that you're talking about, or AB, was already available when Rebecca was first doing these analyses. What causes it's actually very complicated geochemical events that cause serpentinization, which is a geological process in the lithosphere underneath the surface at the vents. And if there's a lot of serpentinized rock at hydrothermal vents, you're going to get a lot of hydrogen. It's an abiotic process.
Okay, so we're actually out of time for the Q&A. So all the questions are going to be moved to Slack. And Nicole, if you could find the time to answer them there, that would be great. Thank you again for a wonderful talk. And we will have a 10-minute break.